Welcome to Module 5.2, Translation and Transcription. This material is covered in Chapter 15 in your textbook. If you recall, we are calling Module 15 the central dogma of molecular biology. That's because we're talking about the way DNA is transcribed into RNA and then translated into protein. Last time we talked about how DNA is replicated. Today we're going to talk about the process of transcription into RNA and translation into protein. So the basic principle here is that genetic instructions written in the language of DNA, which is just G, T's, A's, and C's, directs the formation of proteins and also a whole organism. So the way this works is that genes, which are carried on a chromosome, like we talked about last time, are linearly organized instructions for making RNA, and that RNA is the instruction for making protein molecules. So the structure and function of cells are determined by the thousands of different kinds of proteins that the cells make. So the proteins are mostly what do things in the cell, but each protein, of course, is made up of a primary sequence of amino acids. So a protein's function is based on its shape, folded in 3D, and the charge variations throughout that shape. However, genetic information is based on a sequence of nucleotides, and that sequence of nucleotides determines the sequence of amino acids, and the way the amino acids work together determines how they fold into the final protein shape. So here's the big picture that we're going to be talking about throughout the next few slides. Here you can see that DNA, shown in the blue backbone, is being transcribed by a protein called RNA polymerase into RNA. So those instructions on DNA are transcribed into RNA. And then the RNA is processed. We'll talk about RNA processing. And finally, the end result of a processed RNA is mRNA, which is called messenger RNA. Messenger RNA leaves the nucleus and is then translated into protein through a machinery we call the ribosome. So we'll talk about each of these three big ideas over the next couple of slides. So what are we talking about when we are actually referring to a gene that is translated into a protein? Well, we're talking about a segment of DNA that contains the information required to synthesize one protein. So our genome is a collection of these large chromosomes, and if we zoom in further, we can see that the chromosome is a linear sequence of nucleotides, and a portion of that linear sequence, however long it might be, is called a gene. So let's imagine this is 200 nucleotides long. This gene will encode for one specific protein. So again, a gene is just a block of DNA that encodes for a protein. So the term genome refers to all of the genetic information carried by DNA in a cell, whereas the word gene refers to the information that encodes for one single protein. So the genome contains all the information that's necessary for the synthesis of all types of proteins in your body. But we have a problem. DNA is in the nucleus, surrounded by a bilayer membrane, while proteins are synthesized in the cytoplasm, which is the rest of the cell. So the question is, how do we get the information encoded in DNA out to the cytosol so that that information can be translated into a protein? And the answer to that question is RNA. RNA is the intermediary between these two molecules. So here again is the central dogma. We have genetic information encoded in DNA that's copied or transcribed into RNA and then translated into a protein molecule. So RNA is what moves from the nucleus to the cytosol to take the information in DNA to the ribosome to make our protein. So here's that first big idea we were talking about, which is transcription, going from DNA to RNA. And that starts by unwinding our DNA double helix, much in the way that we unwound the double helix for replication, except instead of DNA polymerase, we have an enzyme called RNA polymerase. So RNA polymerase unwinds a short section of our DNA double helix. Again, it's just going to unwind a short section because there are a lot of hydrogen bonds here and it takes energy to break those bonds. We might as well just only break a little bit at a time. So it unwinds our double helix into two separate strands of DNA. So the 3' to 5' direction DNA is the template strand. The template strand is complementary to the RNA that's being synthesized. The other strand is called the non-template strand, which runs the opposite direction, 5' to 3'. So RNA is synthesized the same direction as DNA, from 5' to 3', which means the template strand is read in the opposite direction, from 3' to 5'. We add ribonucleotides to the growing RNA strand through the action of RNA polymerase, and base pairing is very similar to DNA in that G's always bind to C's, but instead of A's binding to T's, in RNA we have T replaced by U, which is uracil. So the newly synthesized RNA strand, which is shown in this figure with the pink backbone, is complementary to only one strand of our DNA, and it's complementary to the template strand of our DNA. So this, what we were just talking about, what is transcribed by RNA polymerase, is called the primary RNA transcript. This consists of two types of information. We have coding sections, which are called exons, and non-coding sections, which are called introns. So we need to remove the introns before we export the RNA. So the first thing that happens in RNA processing, which occurs inside the nucleus, is called splicing. Splicing is the action of removing the introns and leaving the exons. So if you look here at this top figure, we have an exon and an intron and an exon, etc. What you see here after splicing, we have removed those two introns and we're left with a single RNA composed of those three exons that we had. After splicing, we have two additional things that happen. The first is called capping, which is the addition of a methylated guanine to the 5' end. We call that the 5' cap. And the second thing that happens is called polyadenylation. This is the addition of a series of repeated adenine nucleotides to the 3' end. So it's just A on the 3' end of our RNA. This is called the poly-A tail. So after the process of splicing, capping, and polyadenylation, we now have the final product of our RNA, which is called mRNA, or messenger RNA. And this is what is going to be sent out of the nucleus into the cytosol. But we're not going to let all of the mRNA out of the nucleus. Only correctly processed mRNA is going to be allowed to leave the nucleus out into the cytosol where it can be turned into a protein. If we were to let improperly processed RNA out of the nucleus, we would get an improperly coded protein which would not function correctly. So what we have are specific signal proteins. We call these export factors. You can see them as these oblong shapes here. These bind to our mRNA. Again, we have our mRNA with a 5' cap, 3' tail, and our exons in the middle. This is the information that will tell us how to make a protein. Once these export factors bind to that mRNA, it's determined to be export ready. So at this point, it can pass through a nuclear pore. The export factors will dissociate, which means they will unbind, and what we have is an mRNA outside the nucleus. And once we have an mRNA outside the nucleus, we can start the process of translation into protein. Now we run into something called the coding problem, and that is the question: how is the information in a linear sequence of nucleotides, which is our mRNA, translated into the linear sequence of a very chemically different subunit, which is the amino acid? And the solution to this is a sequence of nucleotides, and mRNA is read consecutively in groups of 3 nucleotides. We call these groups of 3 nucleotides codons. So 3 nucleotides in a row is called a codon. We have 4 types of nucleotides, G, C, A, and U in our RNA, and that gives us 4 times 4 times 4 equals 64 possible combinations of our 3 nucleotides. Each one of these 3-letter combinations is specific for one amino acid. But we only have 20 different amino acids, so we have some redundancy here, which is to say that some amino acids are specified by more than one group of 3, or more than one codon. So, for instance, a specific amino acid will have several different codons that all code for it. So this figure shows the rules by which the nucleotide sequence of a gene, through mRNA, is translated into the amino acid sequence of a protein. So if you look over here, let's pick an amino acid at random. Let's look at proline, PRO. This is encoded by these 4 different codons, CCU, CCC, CCA, and CCG. So when the translation machinery sees any of these 3 nucleotide sequences, it is going to know that the next amino acid in the chain is going to be a proline. But this brings up another problem. So in the process of translating, the mRNA is being read from 5' to 3' in these sets of 3, which we call codons. But how will the amino acids know to come to the mRNA? So we have amino acids sort of floating around in the cytosol, and we have an mRNA there with these codons specifying a specific amino acid. But the question is, how does that amino acid get brought in to the mRNA? So the answer here is a molecule called TRNA, or transfer RNA. So transfer RNA has 2 regions. One region is called the anticodon. That is the part of the tRNA that binds to the codon of mRNA. So this is going to bind to mRNA codon. So the 3 nucleotides here are going to be what we call complementary to the mRNA codon. So the G would bind to a C in the mRNA, and these A's would bind to U's on the mRNA. The other end of the tRNA molecule, if we look up here, has an amino acid attachment site. So here we would have the amino acid phenylalanine. So you can set the codon for one of the codons for phenylalanine must be UUC. And if we look back a slide, you can see, oh yeah, there it is. UUC is one of the codons for phenylalanine. So we have a UUC on the mRNA, which we have a complementary anticodon on this tRNA molecule, which again is not an RNA molecule that codes. It is a functional RNA. Its function is to one end to bind to mRNA and one end to bind to protein, in this case phenylalanine. That way it brings the correct protein to the correct site on mRNA. So this is how the message is decoded. In the cytoplasm, mRNA brings the message from DNA. Then we have the tRNA molecule that we just talked about with 2 binding sites. The anticodon region of tRNA binds to the mRNA and the other end of the tRNA binds to the amino acid. So we have an amino acid bound to a tRNA, which binds to mRNA. We then have protein synthesis, and this requires a machine that will do a couple of things. Number one is move along the mRNA, capture those tRNAs and hold them in place, covalently link the amino acids together to form the protein chain. And you'll remember the covalent bonds between amino acids are called peptide bonds. And the machine that does this is called the ribosome. So the ribosome moves down the mRNA from 5' to 3', grabs the tRNAs, holds them in position, and creates the peptide bonds between our amino acids. So the ribosome has two subunits, a small subunit, seen here below mRNA, and a large subunit, seen here above the mRNA. So this begins, in step 1, with the initiator tRNA anticodon recognizing the codon of mRNA. So the initiator tRNA is always methionine, M-E-T. So the start codon, we call it, is A-U-G. So the tRNA molecule is complementary to that codon in its anticodon region, so it has a UAC. So those two bind together through complementary base pairing. The tRNA brings in the methionine. Second, the large ribosomal subunit comes in, binds to the small subunit, and a second tRNA is recruited. Now, what second tRNA is recruited? By the second codon, which in this case is UUC, which remember is phenylalanine. So we have a tRNA shown here in blue with an anticodon that binds to UUC and the other end bound to phenylalanine. Now you see the ribosome has created a peptide bond shown here by a black line. And that peptide bond has been created between methionine and phenylalanine. So then the mRNA is moving relative to the ribosome, and this process repeats itself. So the next codon would be CGA. Complementary anticodon is GCU. So this tRNA brings along arginine to the large ribosomal subunit, and the ribosome is going to move down from 5' to 3', continuing to read codons, bring in tRNAs, and create peptide bonds between amino acids to form a polypeptide chain, which is also called a protein. So that is the whole process. That is what we call the central dogma of molecular biology. DNA is split apart by RNA polymerase. RNA polymerase reads one strand of the DNA, called the template strand, transcribes DNA into RNA. That RNA is then processed through splicing, the addition of a 5' cap and a 3' tail into mRNA, which leaves the nucleus, moves into the cytosol, and is then translated from RNA to protein by the ribosome. And again, the ribosome brings tRNAs, which are complementary to the mRNA, in, locks them in place, and makes those peptide bonds between the amino acids to form a protein. So this is the entire process. We'll talk more. We'll talk about this in more detail in class, but I appreciate your attention, and thank you for watching.